All right, in this presentation, we're going to be talking about pedigrees and special heredity. Well, a pedigree is a way to follow a trait through a family. And basically, what I like to think of this is a family tree of genetics. And over on the very beginning of this, I sort of show the key on what we're looking at. Now, one thing I do want you to know, affected is a very different word from infected. So please get that straight, that affected and infected are very different words, and they don't mean the same thing. So if someone is shaded on the pedigree, that means they are affected. By being affected, that just means for whatever your pedigree is following, whatever trait it's following, they are showing that trait. That means they are affected with that trait. They have it, they're showing it. Whatever it could be. You could be following hair color, you could be following skin color, eye color, hand size, whatever you're looking at, they have that. So let's look at a version right here. The circles represent females, the squares represent males. A line between them means they had babies, so they had 1, 2, 3, 4 kids, and they all had kids. Now, up here, we're seeing that this is a pedigree showing dominant inheritance, and whatever trait they happen to be looking at, this person had it, this person had it, this person had it, this person had it. And we can look at this and try to figure out what genotypes people have. Now, since this parent doesn't have the dominant trait because we're looking at dominant inheritance, their genotype has to be homozygous recessive. Well, combining them with this parent, we can use a Punnett square to figure out what their kids could be. And by looking at their kids, we can figure out what genotype this parent is. Well, this kid's showing the dominant trait, this one is, and this one is not. The only way these two parents could have a kid that's not showing the dominant trait with one parent that's homozygous recessive means this parent has to be heterozygous. And you can do that all the way down. I'm just using simple Punnett squares. Here's a pedigree that shows recessive inheritance. Now we see carriers that are half shaded. Well, since this is recessive inheritance, are they showing the dominant or the recessive trait if they're not fully shaded in? They're showing the dominant trait here, dominant trait here. All of these people are showing the dominant trait except for this one. This one is affected on this pedigree, and they are showing the recessive trait. Everyone else is showing the dominant trait, even the carriers, because they're heterozygous, they have one dominant allele, they're going to show that dominant allele phenotype. Now, this provides one of the complications with Mendel. One of the big complications with him is most families don't have enough kids to actually see the correct ratios that Mendel provided. Now, your chance of having a boy or girl is about 50-50. 1 out of every 2 kids should be a boy or girl. But everyone knows a family that has like 4 male children or 5 male children or 6 sisters, something along those lines. So because we have a limited amount of kids, probability over a long haul, probability usually gets to where it's supposed to be. But if you're just taking a small sample size, like 3 kids or 4 kids, you might not see Mendel's traits. Another issue with Mendel is that not all traits follow Mendelian genetics, unfortunately. It was a good start for us, but it doesn't always follow things. Another big problem with Mendel is that sometimes mutations occur, and mutations are going to change alleles. And when alleles change, that's going to mess up Mendel's numbers a little bit. When we talk about traits and characters in the wild type allele is the one that you most often see in the phenotype. So that's kind of the most common one. You look at a human, wild type allele for us is having 2 arms. Most humans are born with 2 arms. That's the wild type. There are some mutations that take that away. And when you look at a trait where less than 99% of the people have a specific trait, take hair color, eye color, skin color, then we say that that trait is what we call polymorphic, meaning there are many forms to that trait. Some different forms of dominance, other different forms of dominance that we see that cause issues with Mendel's work is incomplete dominance. With incomplete dominance, a heterozygous individual is going to show a blend of phenotypes between a dominant parent and a recessive parent. So a heterozygous individual is going to show a brand new phenotype or an intermediate phenotype that's different than the dominant or the recessive. I'll show you an example of that in a second. With codominance, a heterozygous individual is going to show both the dominant and the recessive in their phenotype instead of one or the other. So they're codominant. They're both showing up. Here's an example, classic bio example of incomplete dominance. You have the red tulip and the white tulip. When you make them make babies, if they're homozygous dominant and homozygous recessive, they come out with a bunch of heterozygotes and they're all pink. And if you put those two heterozygotes together, well, they have kids too, in the same 1-to-1 ratio of 1 homozygous recessive, 2 heterozygous, 1 homozygous dominant that you should get. But instead of just seeing 2 phenotypes, you'll see 3 different phenotypes in their children. Another issue is that sometimes we have genes that have multiple alleles or more than 2 alleles for a specific trait. A classic example of this is blood type. In blood type, humans possess A alleles, B alleles, and O alleles. Now here's kind of the crazy thing with blood type is not only do you have multiple alleles where you have 3 alleles, A and B are co-dominant with each other. So your blood type can either be A, which is a dominant blood type, B, which is a dominant blood type, AB, which is a co-dominant blood type, or blood type O, which is a completely recessive blood type. And here are the different genotypes that accompany the A blood type, the B blood type, the AB, and the O. Now it's really crazy when we talk about giving blood from person to person, because giving blood, you have to match the blood types. If you don't match a blood type, the blood will clot inside the person's body, and that would kill them. That would be bad. So we try to make sure we know blood type before we give someone a blood transfusion. Another issue that causes trouble with Mendel and his ratios is something called epistasis. Epistasis occurs when you have the phenotypic expression of one gene is controlled by another. Common example of this is in Labrador retrievers and dogs. The dominant phenotype would be the black lab. Black is going to be the dominant one. Brown or chocolate Labradors would be the recessive phenotype. Now, having the dominant or recessive, those things work pretty much exclusively each other unless there's also something called the E pigment. If a dog is homozygous recessive for the E's, then they don't show the black or the brown. Basically, there's no deposit of that pigment on the hair color, and that creates a blonde or a yellow Labrador retriever, what we call the golden retriever. So anytime you see a dominant E in a golden retriever's genotype, they're going to follow whatever the B's are. If it's homozygous dominant B's, they'll be black. If it's heterozygous B, they'll be black, because black is dominant to brown. If they have any dominant E with a homozygous recessive B's, they're going to be brown. No matter what the B combination is, however, if they are homozygous recessive for the E, they don't deposit that brown or black color onto the hair, and they're going to be that yellow Labrador. So when we say that the E is epistatic to B because E is going to be the one that determines the phenotype. So the E is going to tell the brown and black whether they're actually turned on or not. And this has to do with gene expression and all that. That's a topic we're going to be getting into in the next couple chapters as we talk about protein synthesis and gene expression. Here's a close-up of the heterozygous dihybrid cross that we see with a chocolate lab or a black lab that's heterozygous with heterozygous E's, and this is sort of follows that 9331, except that one is now a yellow one, as are these 3. So we only see the 3 phenotypes. Really, we're just looking at the hair color phenotype. So we're really only looking at one as opposed to 2 different traits.